So now let's take a close look at the processes involved with cell respiration. That'll lead us into glycolysis, the citric acid cycle, and the electron transfer chain. And it also will take a look at fermentation, that of lactic acid and of alcoholic fermentation. So with glycolysis, one of the things to know is that, again, with the equation for cell respiration, oxygen was a major component of that. We needed oxygen and the glucose to do things. It's important to note that with glycolysis, this does not happen in the mitochondria at all. So oxygen isn't a part of this equation. So this will happen whether there's oxygen, aerobic respiration, or without oxygen, which is anaerobic respiration. This will happen in either one. So this is happening in the cytoplasm as the glucose gets brought into the cell before it's broken down and put into the mitochondria. And so we start off with a glucose molecule, right? And it's going to go through a couple of steps, and eventually it's going to become two pyruvates, right? Well, the first thing that has to happen is we have to put a little energy into the system. We've got to put 2 ATP in. And of course, 2 ATP in, and that means 2 ADP and a phosphate come out. The next thing we need to do is we're going to take off a couple of these hydrogens. If you remember from our overview, ATP and NADH, 2 of each, was the product from this, in addition to the 2 pyruvates. So we're going to have 2 NADs come in, pick up hydrogens to become 2 NADHs. NADHs will leave and go into the mitochondria. And go dump off that load at the ETC. The last thing that happens is at this point we haven't produced any ATP. We've used ATP, but we haven't made any. So at this point, 4 ADPs and phosphates come in and become 4 ATPs. And then at the end of this process, we end up with 2 pyruvates, 2 3-carbon molecules that are now in a form that can be transferred into the mitochondria, which then later be broken down by the citric acid cycle, the Acetyl Coa cycle and the citric acid cycle. But notice that I said on this one that we ended up with 4 ATP. If you go back and look over the overview, it says we ended up with 2 ATP. So you know kind of what's going on there. We just have to do the math. If it takes 2 to get started, so we burnt 2 already. When we produce 4 at the end, our net production is going to be 2 ATP. So that's how we came up with that number of 2 ATP production for this. So 2 NADHs and 2 ATPs are produced. The end result being that we end up with 2 pyruvates. Each will then now go into the mitochondria. But keep in mind, this process does not require oxygen, and it still produces some energy. Just 2, but hey, 2 is better than none. So glycolysis is actually going to end up being our starting point when we talk about your body's plan B for getting energy when there's no oxygen, which is fermentation. So we're going to see a diagram similar to this a little bit later on. But let's imagine we're good to go. We got oxygen, so we're going to go into the mitochondria. So that the first thing that's going to happen is we're going to start running through the Acetyl Coa cycle, and then eventually into the major part, which is the citric acid cycle. And the whole point of this is to strip down all of the hydrogens off of this chain of carbons and oxygens and start getting ready to release the energy that's stored in each one. Again, because this is inside the mitochondria, this can only occur when there's oxygen. If there's not enough oxygen, this process does not occur. It comes to a screeching halt, okay? So the first thing that's going to enter from glycolysis comes our 2 pyruvates. Now, technically, this process would happen 1 pyruvate at a time, but I think I've added the numbers to kind of help us with to keep up with the 2 pyruvates. I prefer to think of this as a per glucose molecule. So I kind of do everything with the idea that we're looking at the products from one entire glucose molecule. So we have 2 pyruvates coming in. First thing that's going to happen is NAD is going to come in. It's going to start stripping off some of the hydrogens to make NADH. So we got 2 of those, 1 for each pyruvate. That's going to leave and go off to the ETC. But once we start taking off those hydrogens, we don't need all the carbons, and we don't need all the oxygens that are left. So what happens next? Well, a little bit of CO2 is produced. Just a little bit, 2 CO2, 1 from each molecule. That's exhaled. That diffuses out of the mitochondria, diffuses out of the cell, ends up in the blood. It gets exhaled for us through respiration into the air. So we're good to go. But once we remove all that, essentially we're left with just 2 carbons per pyruvate. That's what you got depicted here. In order for this to start off in the cycle for the Krebs cycle, for the CAC, the citric acid cycle, there's an enzyme called Acetyl Coa that comes in that helps prep it for the next stage in its journey, and that cycle is called the Acetyl Coa cycle. It's a big process. It does some cool things, but essentially, for the level of the course, it's not something we're necessarily going to focus on. So once we get those 2 carbons into the Krebs cycle, into the citric acid cycle, the first thing that's going to happen is we're going to remove all the carbons. We simply don't need them anymore. Remember, the whole point of the carbons was to stabilize the hydrogens for storage. We're not storing it anymore. We're going to use the hydrogens, so we don't need the carbons. So as before with the carbons, we attach them to some oxygens and exhale them. Same thing happens here. But we're going to produce 4. That's 2 from each pyruvate, for a total of 4. So the majority of the carbon dioxide that we exhale actually comes from the citric acid cycle, aka the Krebs cycle. But some of it does come from the Acetyl Coa cycle, so I like to mention that as well. But see, now we've gotten rid of the carbons, so we're left with the hydrogens. But we need to start doing other things with them. The first thing that happens is we need to move them, we need to transport them, right? We got to get these out of here. So NAD comes in, 6 of them come in, grab 6 hydrogens, pull them off to become NADHs. That NADH is going to go off to the electron transfer chain, same place the 2 from the Acetyl Coa cycle are going to go. So those head off, all right? In keeping this cycle going, we actually are going to take a little bit of ADP and we're going to transform that into ATP. And this is where the ATP in the citric acid cycle is actually produced. Just a little bit, but it's some. But this, again, it's occurring in the mitochondria, so all this would stop when there's no oxygen. And the last step is we still have a few hydrogens that are left on the remaining molecule. So we need to strip those off. But we've pretty much used all of our NAD at this point, and the structure that's left doesn't bond really well with NAD anymore, so we have a new molecule that comes in that can grab 2 hydrogens per molecule, and that's going to be our FAD. FAD comes in, grabs the remaining hydrogens, pulls them off 2 at a time, and we get 2 FADH2s. Whatever's left, the remainder of that molecule, is now ready to bind to whatever this acetyl A is carrying over from the earlier cycle. That 2 carbon molecule can now bind to it, in which case it will go through the cycle again, and we just call this the citric acid cycle. Again, it's also called the Krebs cycle. I just call it the citric acid cycle or the CAC, but either term is good. So again, we've got carbon dioxide leaving the body, we've got 2 NADs from here, 2 NADHs from there, we've got 6 NADHs from here, and then 2 FADH2s from over here. So we've got a lot of hydrogens being stripped off. Again, there's nothing left at this point. There's no more glucose, there's no more pyruvate, it's all gone, all the hydrogen are pulled off, the carbons and oxygens are being exhaled back out. So yeah, we've pretty much broken down at this point. We have broken down that glucose molecule, which is exactly what we want to do. Now we've got to isolate that hydrogen, get the electron off of it, and put that energy, the excess energy, the photon from the sun, we got to put that energy to good use to generate some ATP for us. So that leads us to the ETC, fermentation.